So today we're going to do two different FRQs. So we're going to do the 2016 number 1 as well as the 2016 number 8. So 2016 number 1 has to do with graphing, a little bit of genetics, and then a little bit of tonicity. So they give you this diagram. They show you what the Long Island Sound looks like. They talk about how this is increasing in salinity, meaning it becomes more salty as you get closer to the Atlantic Ocean. And then they give you these different sites, and they label the sites on the graph. Notice these sites are not equidistant from each other, and they're just specific sites that we see in Long Island. And so they show you this lap frequency. So there's a certain gene, and they show you the frequency of that gene. And so they then go on to explain some stuff to you. They say that laps are found in these organisms. They're associated with a certain muscle, and it has to do with changing in salinity. These are enzymes that remove amino acids from proteins and release these free amino acids into the cytosol. So that means I'm going to have an increase in the concentration of these amino acids in my cytosol. So to investigate the evolution of the laps in the wild populations of these mussels, they sampled a various number of mussels, and then they gave you, of course, the data that we see in our table. So the first thing we want us to do is construct an appropriately labeled bar graph to illustrate the observed frequencies of the lap allele in the study populations. So sometimes they'll tell you exactly what kind of graph to make. And I've seen tons of students who make the wrong graph. So if they say in the prompt what kind of graph you need to make, go ahead and circle it, underline it, do something to remind yourself what kind of graph it is you're going to make. Now, the information I've been given is the sites, the frequencies, and then salinity. So what I'm going to do is I'm going to put the sites on my x-axis and my frequency on my y-axis, because depending on the site is where I'm seeing a difference in that frequency. And I go ahead and graph all my points. So the 3 points you got for this was correctly plotting your bar graph, correct axis labeling, so having the sites, having your frequency, and then of course scaling and our units. So students oftentimes mess up with scaling. Notice that the scaling has to do with the number of little blocks we have. So every 3 blocks equal 10. And so you want to make sure that you have an equidistance between them all and that each block means the same amount in terms of your values. So based on the data, describe the most likely effect of salinity on the frequency of the allele in the populations. So what we see is that as the salinity increases, we're seeing an increase in our lap allele. And so that's what you need to talk about. You need to say that as the salinity increases, the lap frequency increases. Now, a lot of students will say this backwards, and they'll talk about that as the frequency increases, the salinity increases. That is not appropriate. We cannot say that because it's not the frequency that's causing the salinity, it's the salinity that's causing the frequency. And so you need to make sure you're understanding the causation. You could have also said the opposite, where as salinity decreases, the frequency also decreases. They then ask you to predict the frequency of the sampling site between 1 and 2. So 1 was at 13 and 2 is at 16, if I remember correctly. And so what they wanted you to do is they wanted you to either give a point, any point you want between 13 and 16. You can predict a point. Or if you're going to give a range, you have to give it based on your known. So you know it's somewhere between 13 and 16, but you don't know the actual value. You could just say, I predicted something between 13 and 16. Or again, you could have given a specific number, 14, 15, 14.5. Any of those numbers would have been appropriate. So the student that did this, they went ahead and wrote out that we see the increase in salinity is associated with an increase in the frequency. They also then said that it was going to be 15%. They picked a certain value between those two. So C says that we need to describe the most likely effect of the activity of osmolarity on the cytosol, and then describe the function. So when we're thinking about our lap activity, we're going ahead and we're talking about how increasing of that activity is going to increase the amount of amino acids. And so the more amino acids I have, the more concentrated my cell is, and thus the more hypertonic it is, and the lower the water potential. And so you talk about how increasing the lap allele increases osmolarity or the solute concentration, or you could say that it decreases that water potential. We also talk about describing the function of the lap on maintaining the water balance. So if the inside of my cell becomes more hypertonic, it's going to inhibit the water from being lost. If our environment is hypertonic and my cell is hypotonic, water constantly will be leaving these cells. And I need my muscle cells to maintain their water, otherwise they're going to get dehydrated. So by increasing the concentration inside of the muscle, it makes it so that the muscle doesn't lose water to its surrounding environment. So the student talks about how it releases these amino acids that lowers the water potential. They then go on to talk about how, because we have a hypertonic environment surrounding the cell, that would cause the water inside the cell to leave, which then, of course, is allowing them to offset that balance. So D talks about that our marine mussels are evenly distributed throughout the sample area. As larvae mature, they attach to rocks. So explaining the differences in that lap allele among those adult mussels, predicting the distribution, and of course, justifying it. And so if we find that they're in a very high salinity area, those mussels that have a higher concentration, a higher frequency of the lap allele are going to be more likely to survive. And then they're not going to be as likely to survive in the hypotonic or the low salinity environment. And so that kind of gets us a little bit into evolution on the causation of more alleles means that I'm going to be more favorable in a high salinity environment, which of course that connects. Predicting, we're going to see that mussel populations will increase in that high salinity area, or the mussel population will decrease in that low salinity area because of the fact that they have the higher amount. So they're going to be able to survive in that high salinity area. And so those ones will be able to osmoregulate. So the person talked about how the higher populations of individuals would then survive in those areas of high salinity. They talk about greater numbers of mussels in the higher areas of high salinity would then, of course, have that lap allele. And so that brings us to the end of 2016, number 1. So now let's talk about experimental design and error bars. So we look here and we see that we're looking at exercise, and we see that there's this blood plasma prolactin, and we're seeing that at time 0 and at time 1 hour, we're seeing what happens over time. So they said, ooh, I clearly did not separate these. Justify the use of without exercise as a treatment. Well, if I am trying to see what happens with exercise, I need to see what happens without the exercise so that I can provide a baseline. I know exactly what normal looks like to my patients, and I know what normal prolactin levels look like. This also allows me to ensure that any change I see in prolactin has to do with the exercise because I have that baseline, because I know what it originally started at. So it says to see what normal levels of prolactin are without exercise. B then says, using the evidence, determine whether prolactin release changes after exercise. So if we look here at with exercise at time 0 and time 1, we do see that it increases with exercise. So at 1 hour, there is an increase. But the problem is these error bars are overlapping. And because the error bars overlap, our data is not statistically significant, which means that, nope, prolactin does not increase with exercise. We could also look here at our with exercise 1 hour and our without exercise 1 hour. Again, you see with exercise, there's a higher prolactin level. But again, error bars overlap. And because the error bars overlap, the data is not statistically significant, so we cannot say that exercise increases prolactin levels. And so that is where they were going with that. You had to say the exercise does not affect it, and you had to be very specific on which error bars you use for your justification. So the student talks about how prolactin level does not change. They also talk about time 0 and time 1. Those error bars are overlapping. So I hope that was helpful. Remember, AP Bio Penguins are just success. Bye, y'all.